Hello.
Yes, sir.
Is my screen visible?
Yes, sir.
So last time we did was glycolysis, right? We completed glycolysis. Yeah. So one thing I wanted to change was like, what is the final enzyme that converts pyruvate? Okay. I mean
So
the
pyruvate to acetyl-CoA.
no, like just before pyruvate, like I mean the enzyme that converts into pyruvate.
PEP to pyruvate is converted by pyruvate kinase.
Yeah, pyruvate kinase. Yeah. So one thing I yes, last time I did thing was wrong was mm, pyruvate kinase, right? I like last time I mentioned something, right, which was regulator of this enzyme. What was the substrate?
The regulator was fructose 6 phosphate. It was inhibiting
It was
the
not
enzyme.
fructose 6 phosphate, it is more fructose 1,6 biphosphate. This phosphate. So just change that. I forgot to mention that. Okay. Now from glycolysis. So this was the final thing about glycolysis. Now from glycolysis, what we get is basically pyruvate, and in which two molecules of ATP is consumed, 4 ATP is gained, and we get how many molecules of NAD?
One.
No, okay, it's two, right? Two, because like after glucose we get glyceraldehyde 3 phosphate, and that both, and this is DHA, right? I forgot what was that.
DHAP dihydroxyacetone phosphate.
So same thing. So this will like basically twice the thing will work. That's why we are getting 4 ATPs and 2 NADH and 2 pyruvate, right? So okay. Well, I have to add something planned for this thing. Okay. So like for like next step to glycolysis is TCA cycle, and then it was oxidative phosphorylation. So this took for this entire step to occur, what we need. I think I mentioned two things, right, which is most important.
One is oxygen and then mitochondria.
One is oxygen and mitochondria. So we know that glycolysis occurs in cytoplasm. Where does TCA cycle occur?
TCA cycle is further oxidation of pyruvate to carbon dioxide, making more reduced form of electron carriers
And like where does it occur?
in the matrix.
In the matrix of mitochondria.
Mitochondria.
Okay, let's think. So basically, so what is the general, like what is the shape of mitochondria?
Like oval shape?
Can it be any other shape? So mitochondria is like not just oval shape, it can be coiled, it can be fragmented, it has a lot of shapes. It's not just oval. So like it has like different shapes, like I think based on different organisms, I guess. I don't remember exactly. So this is the matrix. So here our TCA cycle occurs, right? Okay, TCA cycle. Hmm. So the final product was pyruvate, that is a 3 carbon molecule, pyruvate. So now this pyruvate has to go inside the, so this is the cytoplasm, and this pyruvate has to go inside the matrix. So can it go directly? So this is the outer membrane, this is the inner membrane, this is inner space, interstitial space, and this is the matrix. So now we have a, after glycolysis, we have 2 molecules of pyruvate. Now can this pyruvate directly go into the matrix?
No, sir.
So like, is there any special transporter for it? Okay, like first thing, like out of like we have 2 membranes, right? Like we have outer membrane and we have inner membrane. So which do you think is much more permeable compared to, like, I mean, out of these two?
So I think outer membrane is more permeable.
Yeah, outer membrane is more permeable. So this pyruvate can, like, I mean, most of the pyruvate can easily diffuse into the, I mean, like interspace, this inner space. Now from this inner space, when this pyruvate comes here, now from this inner space, it has to go inside the matrix, and like for this we require a special transporters for this. So like there is a transporter called Tom and Tim. Have you heard of it?
Yes, sir.
So what is Tom?
Sorry, sir. Can you please repeat?
What is Tom? Tom and Tim. So what is
Tom is translocon of outer mitochondrial membrane and
yeah. So this is basically translocase,
okay.
and this is like of outer membrane, and this is translocase for inner membrane. So like there are like different types of Tom and Tim. Like it is mostly involved in signaling of, like, I mean, transport of protein, etc. So what this pyruvate, even though like Tom is present outside, it does not need that much. So it easily like it somehow diffuses in, and from this inner space, if it has to cross the inner membrane, it requires the help of Tim. So TIM is the most important thing. Like, I mean, the translocase inner membrane is important for transporting pyruvate from outer, like, I mean, the cytoplasm to inner matrix. So this was the first thing. Next was like, okay, now we have a next thing with, yeah. Hmm. So now when this pyruvate comes into uh, this matrix, it is converted into acetyl CoA, and then there are like a number of cycles. So this is what which cycle it is? Krebs cycle, right?
Yes, sir.
Why do we call this a cycle? And like for example, we don't call glycolysis a cycle, but we call this as a cycle. Why do we do that? Like, I mean, do you have any idea about that?
So maybe because the intermediates of that cycle are recycled like oxaloacetate.
Yeah. So we, in metabolic terms, anything that, like for example, the first product and the last product, if they are same, that means it basically forms a cycle. Like yeah, it gets regenerated. So basically, oxaloacetate, it basically like is utilized and then again regenerated. So okay, pyruvate. Pyruvate is a 3 carbon molecule. What about acetyl CoA?
2 carbons.
So 2 carbon and then a CoA thing is attached to it.
Coenzyme.
Okay. Okay. So if I ask you to define like Krebs cycle, so like how will you define it? If Krebs cycle is basically a reaction of what? Like two major reaction keep like the basically whole Krebs cycle is basically like consisting of two reactions, like two major reactions that's happening. Um, can you guess that?
First one is decarboxylation,
Yeah.
and other is, I guess, removal of hydrogen,
Yeah.
dehydrogenation.
Dehydrogenation. So basically, entire Krebs cycle can be like summarized into that. Basically, it's a process of like it's a cycle of decarboxylation and dehydrogenation, and then so basically CO2 is released, and from this CO2 then dehydrogenation occurs, right? So what is the dehydrogenation process here? So dehydrogenation process is conversion of NAD into NADH. Basically, oxidation reaction is occurring here. Sorry, dehydrogenation, and then so in this Krebs cycle we get NADH, and then we also get, so like we all know that, right, NADH is when it is converted it gives us 3 ATPs. So there is one more energy currency here. Like what is that?
FADH2.
FADH2. So FAD is converted into FADH2. It is not the conversion of this reactant into the product. It's not that energy giving. So that's why like FADH2 is produced, and this FADH2 is like compared to NADH, it is of minimum energy. So if this FADH2 is, like if it goes down to this oxidative phosphorylation, it will give only 2 ATPs. So we know that. So any other thing like comes here? Is there any?
GTP.
Huh?
GTP, so GTP or ATP.
So basically GDP is converted into GTP, and this GTP is utilized to convert ADP to ATP. So this is a substrate level phosphorylation. So basically we get one GTP which is gives one ATP here itself. So this was the entire cycle, right? So basically 2 CO2 is released, and then NADH, FADH2 and GTP is produced. So this is the entire like summary of the Krebs cycle. Now I have one question to ask. That question. So do you remember that glycolysis was regulated mainly by hormones?
Yes, sir.
What were the two hormones?
Insulin and glucagon.
So what about Krebs cycle? How is it regulated? Do you think hormones influence this?
So I don't know. I only know about the ATP and the products which allosterically inhibit the enzyme.
Okay. So basically, yeah. So when you are mentioning this ATP or like any other product that is inhibiting this, so it is basically, Krebs cycle is basically dependent upon the energy requirement of the cell. If it requires like more energy, it will like fasten up. If it requires less energy, it will slow down the process. So what are the energy currency here? We already talked about ATP, NADH. So if ATP is like for if the cell has high number of ATP and NADH, that means does the cell require need to like I mean does the cell require to perform Krebs cycle?
No, sir.
No. And okay. So basically, we in terms like I mean we usually focus on this ATP by ADP ratio and NADH by NAD ratio. So if this ratio is high, do you think the cell will need to perform Krebs cycle?
No, sir.
So here there is no Krebs cycle. Well, here Krebs cycle will be performed because this is energy low. So the one thing here is that unlike glycolysis, which is mostly regulated by the hormones, here the Krebs cycle is regulated by this entities like I mean ATP, NADH, ADP, AMP, NAD. So this is basically dependent on the energy requirement of the cell, and these are the energy they are responsible for inhibiting as well as starting this Krebs cycle. So it is not dependent on the hormones; it is mostly on the energy requirement of the cell. Okay. Now, mm, forgot to mention anything. Okay, this was the one thing. Next, okay. Now we have pyruvate. From this pyruvate, we get acetyl CoA. So, like, is this a part of Krebs cycle for conversion of pyruvate to acetyl CoA?
No, sir. It's preparatory phase.
So, proprietary based. Okay. Now, this is two carbon. So, from this, like, from three carbon, it is getting converted into carbon. So, like, one thing we know is CO2 is getting lost. So, decarboxylation is occurring. Okay. Now, can you name the enzyme here?
It is. There are three enzymes. Complex is called as pyruvate dehydrogenase.
There are three enzymes. Three enzymes. The complex is called pyruvate
Pyruvate dehydrogenase.
dehydrogenase complex. Okay. So can you name each of these complex? Let's name it as E1, E2, and E3. So can you name the first enzyme?
Sir, I don't remember their names.
So this is like okay. This has a coenzyme with it that is TPP. Do you know what is TPP? It is thiamine pyrophosphate, I guess. Yeah, thiamine pyrophosphate. And this enzyme was called as okay. Like first, I'll mention all these enzymes. And this is TPP. Next is remember this. This is FAD and NAD plus. This is one coenzyme comes in. There is some you have not studied this parts of this enzyme that are responsible for like conversion of pyruvate to acetyl CoA.
Sir, I have studied and I came with this question only that can you please explain the entire complex and its parts? That was the most challenging thing in the entire website.
So it was something lipo like. One second, sorry. Do you remember the name at least? Like it was something liposilic or lipo something. Pyruvate dehydrogenase. Did you get the enzyme name? Like it was
Yes,
something
sir.
like
In the PPT, it is written lipoic acid, and it is with lysine.
lipoic acid. Can you spell it? LIPO? IC.
LiPoIC. Yes.
IC. Lipoic acid. Yeah. Okay. Okay. Like I'll better write the names of this enzyme as well. So first one was pyruvate dehydrogenase. Pyruvate dehydrogenase. Second is dihydro. So first one is pyruvate dehydrogenase. Second is dihydro lipoic transacetylase NPTEL dehydrogenase. Okay. So this is basically the entire complex, that is pyruvate dehydrogenase complex. It has three enzymes, that is pyruvate dehydrogenase, dihydroxy lipoyl transacetylase, and then dihydroxy lipoyl dehydrogenase. Okay. Now for this, we'll take each enzyme one by one. So first we'll talk about pyruvate dehydrogenase. So what it does is basically, for example, this is an enzyme. This is the binding domain. So what it does is basically it takes up the pyruvate, and this is thiamine pyrophosphate. So this is the the coenzyme which is responsible for the action of this enzyme, pyruvate dehydrogenase. Okay. So it takes the pyruvate in, and then with the help of this TPP, it basically releases CO2 out, and then gives a two molecule, that is acetyl, two carbon compound, that is acetyl, and it is this is with the TPP, like it is bound to TPP, and this from this TPP, this acetyl will go directly to the lipoic acid. So lipoic acid as well is, so this is pyruvate, and it is held by TPP. TPP is responsible for conversion of CO2 is released, and this TPP basically will give us two carbon molecule. This is pyruvate dehydrogenase. So this enzyme E1 converts this pyruvate into acetyl after the release of CO2. Now this TPP is taken, this acetyl CoA is taken by enzyme 2. So it has two cofactors in coenzyme, that is lipoic acid and coenzyme A. So this acetyl is received by the lipoic acid. So it's the lipoic acid that receives this enzyme LPP, receives this acetyl, and when this it interacts with LPP, and then later what this does is basically this LPP, that is lipoic acid, takes away hydrogen from it. This acetyl compound leaves hydrogen atom, basically the protons here, and this acetyl is attached with COA. This coenzyme will go and attach the COA here, and then this hydrogen that is acquired by this LPP, that is reduced now. I forgot the name of the reduced part. It will go to this third enzyme, which has FAD and NAD plus. So this hydrogen atom is received by this FAD, and FAD is converted into FADH2. Now this FADH2 will now help this NAD to get converted into NADH. So basically, when we convert pyruvate to acetyl COA, this entire complex, that is pyruvate dehydrogenase complex, helps in conversion of pyruvate to acetyl COA. So wherein there is a release of CO2, that is, you can be seen here, which is done by pyruvate dehydrogenase, and then we also have this NAD is converted to NADH. So this was the entire process. Like, I mean, when pyruvate is converted into acetyl COA, this pyruvate dehydrogen complex releases one CO2 and then converts NAD plus to NADH. So these are the enzymes mostly. Like mostly people will target, like, I mean, this conversion of pyruvate to acetyl COA is also like very regulated step, in which this particular enzyme is highly regulated. So most of this, like, I mean, like most of the positive and negative regulation is targeting this particular enzyme. Like, I mean, this particular enzyme of the entire complex. So did you get it? Like, I mean, this was the like simplest explanation I could give. Actually, I don't remember the exact the pathways.
And this the whole process is it called as substrate channeling?
Huh?
Sub is the whole process called as substrate channeling?
Actually, I never heard about this name. Like, maybe, like, I don't know. Like, I mean, when I read about this, like when I used to read this, like I just remembered this pathway. Like, I mean, I didn't know. I don't know the exact name. Like, maybe. So, like, sir has taught it, like, or you have read it from some textbook or something.
Sir has also taught in NPTEL it is written substrate channeling, so I was a
So
bit
maybe that
confused.
might be like I have I mean, like, I might have forgotten that, but I don't think I have like come across such a term. So like this was the entire process where like these are the three main complexes, and you have to remember this cofactors, like the coenzyme that is responsible for the action of each enzymes. So the TPP, that is thiamine pyrophosphate, and then lipoic acid coenzyme A, and then that is flavin something, and then niacin. So any deficiency of vitamin, that is thiamine or flavin or niacin, can, like, I mean, inhibit this step. So the conversion of pyruvate to acetyl CoA, and then later like generation of much more higher amount of energy is not will not be possible. So like this is well regulated step. So now let's target, like, let's focus on do you have any other doubt in this?
No, sir, this is clear, but I have one general doubt. Can I ask that?
Yeah, sure.
So, sir, actually, we are left with only 4 lectures, and there's a lot to cover because the course is quite challenging, sir. So, I was hoping if we can little speed up and focus only on the difficult concepts like this one.
Okay. Hmm, difficult concept. So sure, okay, we can do that. So difficult concept, like right now, like you guys are like learning photosynthesis, right? Like I mean,
Sir, in photosynthesis, the light reaction was explained very well, but uh, I was confused in the carbon assimilating reactions.
okay.
And also in the entire course, I think the bio energetics part was the most
Yeah,
difficult
like
to
it's
understand.
also like the thermodynamics part. Like, yeah, I understand that. So in bind, okay, like once, like, tell me this thing. So in this photos, like in maybe like next class is on Sunday, right? Yeah. So photosynthesis, you don't want the entire thing. You just want the carbon
Carbon
assimilation part.
assimilation, yes.
And then one left thing is like bioenergetics. Then what specific in bioenergetics?
Uh, so actually, there was a concept about delta G, and then a lot of different delta G's, then
Okay, delta
reduction
is okay.
potential,
I can understand. Then the next.
and then the how large energy, I mean, how the energy rich compounds are hydrolyzed to get energy from them. And then there was one part in the, I think it was in week 5, which was talking about the 5 main reactions which are
Yeah,
predominantly
yeah.
seen in all the biochemistry.
Okay, those reactions. Okay, and then in anything else? Like
And
actually, like I too don't have time. Like I mean, PhD is quite hectic right now. That's why I'm not able to like do this thing properly. Anyhow, like okay, next. What else?
and so can you please provide the solutions for the numerical problems in the assignment?
Okay, solution for numerical problems. Okay, next.
So that's it.
Like after photosynthesis, like you might have like studied that urea thing, right? It's going on, right? No.
Yes, yes, sir.
So
Currently
like
going
you
on.
like there is no thing like no doubt in that.
So I have not started it. I'll be starting it from today and then I'll finish it by tomorrow.
You are a BSc student or a MSc student?
So actually, I have done my BSc and I'm preparing for masters.
So these are the four things. Okay. I'll teach this in like in Sunday class. Like I mean, like I'll be focusing only on these things. The rest of it, like I can ignore. Like if there is something like important, if I need to tell, then I'll just add that. Okay.
And also, Sir has taken up a lot of mechanisms in the class. And do you think they're going to come in exam? Because in assignment, very small questions were picked up from those mechanisms.
Actually, I haven't went through the course. Like actually, I just check out the topic and I just teach. Like I don't have that much time, like due to my experiments. Like, and I don't even know like how these exams are conducted. Like they are like MCQ based, right? I like as long as I know, like, remember.
So I think they're MCQ based, but particularly this course is very. They have made everything complicated. They're giving fill-ups and then numericals and then matchups.
Okay. So like I have no idea. Like I mean, what they will ask. And when is the like exam scheduled?
30 April.
30th of April. And then last session. Okay. Okay.
And
I'll
I
just.
think, sir, in other courses, they're having more teaching faculties,
Yeah,
more
like
than one as well.
this course should have had like 3, 4 faculties. I think. Yeah, I know. Okay, then like on Sunday, like I'll mostly like I'll discuss on this. So I'll cover all this bio. Like I understand that this bioenergetics part will be like quite tough. It's like I mean I know that it's tough because it was for me as well. Um, and then solutions of nucleic. Okay, yeah, okay. On Sunday, like we'll discuss these things like particularly. Now let's finish this Krebs cycle first.
Sir, if there's nothing difficult in Krebs cycle, we can quickly finish the oxidative phosphorylation as well.
Okay, like I have few things to add, like because like I saw the lecture notes. Actually, I didn't see the went to the lectures, but I saw the lecture notes. There are few things that Sir didn't mention, so like I was planning to mention those. So like
Okay,
I'll mention that first, and then we can quickly shift it to Krebs. I mean this that's like what this thing, um,
okay, sir.
the oxidative one. So okay, we were discussing about okay, yeah. So as I was mentioning, this pyruvate to acyl CoA conversion is like quite I mean like regulated step. So in that step, mostly this enzyme is basically mostly regulated in the entire complex. So as I told you that this entire process is not regulated by the hormones, but are regulated by the enzymes. Not sorry, not the enzymes. Um, the energy level in the cell. So so this enzyme will. So this is the positive regulator. This is negative regulator. So first thing is ATP and sorry, these are the negative regulators. Positive regulator is ADP and NAD, and negative regulator will be ATP and NADH. So these are the direct inhibitor of these enzymes. But these enzymes can also be inhibited by two further enzymes. Like um, one is a kinase and one is phosphatase. So like, have you studied about this? Like, I mean, the two enzymes that regulate this enzyme.
No, sir.
This phosphatase and kinase. So basically, this one of the reaction is that ADP and NAD directly can go and like bind, like, I mean, stimulate this enzyme, stating that the energy level of the cell is quite low and we need to process this Krebs cycle much like a lot. But this can also be done by this phosphatase, and this can be like stopped, or like, I mean, it can be inhibited or started by this enzymes as well, that is kinase and phosphatase. So when this kinase, what it does is basically goes and phosphorylates this enzyme, and when this phosphorylation happens, it will basically stop the Krebs cycle. And when this phosphatase gets activated, it will go and remove this phosphate, and then it will start the Krebs cycle. So now, how do we know that this enzyme should be phosphorylated or not? So this again is indirectly stimulated by ADP, ATP, NAD, and NADH. So one stimulation is direct stimulation. They can directly go and bind to this even complex of the enzyme, or it can stimulate this pyruvate kinase and do the same thing. So this ADP phosphorylation is basically stopping. Stopping means ATP and NADH will go and bind. But this phosphatase is not regulated by this thing, it is regulated by the calcium intake. Do you know, like, when do we need this calcium? For example, if it's a muscle cells and we are like, for example, we are exercising or like stretching. So that means we need like a lot of calcium influx happening in the cell, right? The motion of actin and myosin.
Yes. Yes,
So the
yes.
calcium gets in. So what is like, which organ is the source of calcium?
So it is endoplasmic reticulum gated by IP3.
Like more specific?
ARIA.
Calcium is RER. Are you sure? Have you heard about SCR? Not smooth one. Sarcoplasmic reticulum.
Yes.
So this is the calcium containing bag. Like basically this is like mostly prominent in muscles. When you talk about muscle cells, myocytes, you will find SCR there. SCR basically is a bag of calcium. So like when we need calcium, it basically releases, and this calcium goes and activates this phosphatase, and this phosphatase goes and removes this, like for example, when I'm stretching, like I'm exercising, that means that actin and myosin are moving, and that means like lot of calcium intake is done into the cell. Now this so basically the cell is telling me, like, like the cell basically needs much more energy, like much more ATP. So when this calcium gets in, it will go and for like activate this phosphatase, and it will remove the phosphate from here. And then when this phosphate is removed, it basically will give the cell more energy. So basically calcium is regulating the cycle. And in one more thing, in Krebs cycle, most of the enzyme here is calcium sensitive. Like actually, like most of the enzyme, yeah, like all the enzyme are basically calcium sensitive. So basically, calcium can easily regulate this cycle easily. It can easily regulate the cycle. Okay, one question I wanted to ask was like, is Krebs cycle catabolic reaction or anabolic reaction?
So since we are breaking, it's catabolic, but the some of the intermediates are used for anabolic processes.
So basically, it's an amphibolic cycle. Like it basically
Yes.
does both catabolic and anabolic both. Okay. Now let's get into the actual Krebs cycle, which like I'll mostly focus on the uh, reactions. So, like, what's the carbon number of oxaloacetic acid?
4.
So this is 4. So this is 3, and this is 2, and this is 4. So like what's the enzyme that converts, like, I mean, from this oxaloacetic acid to what's the first product that is formed?
Citrate.
Citrate. So what's the enzyme here?
Citrate synthase.
Citrate synthase. So this was the one-way enzyme, like pyruvate dehydrogenase complex. So this is a one-way enzyme. So this is highly regulated. So I have like already told you, like it can be regulated by ADP or ATP ratio, and then NAD and NADH ratio. And then it can also be regulated by which enzyme?
Kinase and phosphatase.
And kinase is regulated, like this is the direct this is the direct regulation, and this one is indirect regulation. Kinase and phosphatase. Indirect regulation. So this is one of the most regulated step of the um, Krebs cycle. What was the most important step in the glycolysis?
So fructose 6 phosphate to fructose 1,6 bis phosphate.
Okay. This is indirect regulation. Okay. Now this is citrate synthase. Citrate is a how many carbon compound?
6.
6 carbon compound. Okay. Now again, since this is a like one-way process, so this thing is also highly regulated, and this enzyme, it has like various binding pocket, and as I told you, like energy level is much more important here. So one thing is like ADP, ATP. Next is NADH. Do you know other two things?
So I think citrate itself is able to feedback inhibition.
Uh, Citrate. I don't think so actually. No, citrate was not there, I guess. Um, citrate is a like inhibitor of which in glycolysis it comes, right. It was inhibitor of which enzyme?
PFK1.
PFK1. But I don't think citrate is an inhibitor of citrate synthase. It is one like complete cycle. Isocitrate, and then what comes?
Uh, Alpha ketoglutarate.
Alpha ketoglutarate, and then is succinate, then fumarate,
So then malate and fumarate.
malate,
Fumarate, then malate, then oxaloacetic. Okay.
fumarate, malate, and oxalacetate. Okay. So now, like citrate is not an inhibitor of citrate synthase. It is this succinate is an inhibitor of succinate CoA, right? This succinate CoA, right?
Succinyl CoA.
Succinate. And then one more, there is one more regulator of this enzyme, that is fatty acyl coenzyme. So basically, when there is like abundance of glucose already present here, and then it has to be it is basically converted into fatty acid. So that fatty acid is also like uh, for storage. During the storage process, it comes and inhibits this citrate synthase, telling that we need the citrate for fatty acid and not like not further conversion, like not like, I mean, there is no requirement of energy. So this citrate, This pyruvate can be converted from like okay. What is the source of acetyl CoA?
Uh, so one is the oxidation of glucose, and then we also have it by breaking down of fatty acids, and then I guess it's also from protein.
Yeah. So these are the three components from which we get acetyl CoA. Okay. This citrate, as you told me that this is both anabolism and catabolism reaction. This citrate is utilized in which cycle? This citrate from matrix, it basically goes into cytoplasm and wherein lipogenesis occurs. So lipogenesis occurs. So like this is one of the, I mean, pyruvate is not just like, sorry, Krebs cycle is not just an energy giving process. It's mostly a anabolic, like anabolic process as well. So first thing is lipogenesis. Then like you guys have learned about gluconeogenesis, right?
Yes, sir.
So like what is the like, I mean, in this Krebs cycle, which is the reactant here?
So is it oxaloacetate?
Yeah, from this oxalic acetate, um, gluconeogenesis occurs. Now, third thing again, like we have lipids, we have glucose forming here, and what is the third thing? There's a formation of amino acids as well from this alpha ketoglutarate, right?
Yes, yes, sir.
So basically, this is the anabolic process of. So Krebs cycle is not just about giving energy; it's all also about formation of various compounds from like from lipids to glucose, and then to amino acids as well. Okay. So this is the one of the like regulated steps. So one is ATP, one is NADH, one is succinyl CoA, and one is fatty acyl CoA. Okay, next is conversion of citrate to isocitrate. It's not what is the enzyme called? Akinase. What was that?
Yes, yes, it was a contest.
Yeah, so like I mean like it's not like it's a two-way process, so it is not that well regulated. Now conversion of isocitrate to alpha ketoglutarate is again one of the main steps. So here first thing is release of CO2, right? And then conversion of NAD into, so when the isocitrate is converted into alpha ketoglutarate, this like I mean the release of energy is packed into is taken up by NADH. So NAD and NADH is converted, like I mean it's formed. So this is one, like what is the enzyme here?
Dehydrogenase.
So again it's a dehydrogenase, like isocitrate dehydrogenase, right?
Yes, yes.
And then next is again one of the most important step, that is conversion of alpha ketoglutarate to succinyl CoA. Here again CO2 is removed and NAD is converted into NADH. So here again one complex comes in, right? What is the complex name?
Alpha ketoglutarate dehydrogenase.
Alpha ketoglutarate dehydrogenase complex. So again it has three enzymes similar to this our pyruvate dehydrogenase complex. It has three complex. They work in the similar fashion. E1, E2, and E3. It's all, everyone has like same thing like TPP. This is lipoic acid and then coenzyme, because like this alpha ketoglutarate is converted into as like succinate coenzyme. So when coenzyme comes in, so it is there, and one is FAD into NADH, NAD. So like similar, like the function is entirely the same. Like what is the, it is similar to that PDC. Now next is like succinate CoA to fumarate. Here like this is like I mean the conversion here is like the energy release here is like very minimal, right? So here the step is GDP to GTP, and GTP here itself is used to convert ADP into ATP. So this is the substrate level phosphorylation occurring here. Next conversion of fumarate to malate is again the energy is not that good. So here, like only a small amount of energy is released. FAD is into FADH2, and then from mallet, I think this is the third place, right, where NAD is released. Or this. Hello.
So I think that FADH2 is formed from like succinate to fumarate in that process.
Like both are occurring on the same place.
Uh, no, there is the succinyl CoA and then we have succinate. So GTP is formed from succinyl CoA to
Okay.
succinate.
Then succinate to fumarate and fumarate to malate, right? Okay.
Yes, yes.
Succinate, succinate to fumarate, malate. So like from succinate it was 80, okay, this right? Then succinate to, okay. So from when conversion of succinate to fumarate, FAD is formed.
Yes, sir.
And NAD is formed here at this place, right?
Yes, yes, sir.
Okay, so like I had to tell you something in this process. What is that? I told you about this. I've told you about this. About this. Okay, like this is the entire. Yeah, this is the entire Krebs cycle. I hope I'm not missing anything. And then again, the oxaloacetic acid is regenerated, and from this, amino acid is formed. And like like do you know this pyruvate can also be directly get converted into oxaloacetic acid?
Yes, yes, sir.
So when the like I mean there is the energy requirement of the cell is like not there. This pyruvate is getting converted into oxaloacetic acid, and then gluconeogenesis is occurring. Similarly, when there is no need of energy, this pyruvate is getting stored into fatty acid, and again this like amino acid formation as well. You can help in that. Okay, now let's sum it up with thing.
So, sir, is pyruvate an apyuritic compound?
What?
Is pyruvate an apyuritic compound?
Hello. Hello.
Yes, sir, now you're audible.
So like you told like what compound?
Is pyruvate involved in anapyrosis? As it was a question asked in assignment.
What is anapyrosis?
Uh, Anapyrosis and catapyrosis is where the intermediates are used to make something else,
Yeah, yeah, like
just like we have seen.
yeah. Then pyruvate is like it is like I mean anapyretic compound. Yeah, because it is used to make this oxaloacetic acid here, right? So like there is like the entire process is dependent on this oxaloacetic acid. So yeah, it is anapyretic compound.
Okay, sir.
Okay, okay. Now let's wrap it this thing up. Okay, I have one few questions. So first question is like normal ATP thing. Like entire like if there is a like a cell which has like all everything, oxygen and mitochondria. So what is the entire ATP output?
So 32 to 36 after electron transport chain.
Hmm. How it's 32, 36?
And if you are asking only from Krebs cycle, then it is
No, no, no. Like
12.
glycolysis plus Krebs plus was that oxidative phosphorylation? The entire thing, the whole process. Like from if I have one glucose molecule after oxidative phosphorylation, what is the net ATP that I will get? Let's go by this. Glucose.
So, okay.
From this
So
we
I
get
think that from glycolysis we are getting 10 ATP.
from glycolysis you're getting 10 ATP.
No, so you said now that we are getting NADH, NADP,
So like okay,
NADH.
let's go step by step. So we are getting two ATPs lost, right? From here like everything's twice. So like we get two NADH like after this. We get two NADH. So from this we are getting six ATPs. Okay, and then four ATP. So basically from this
10.
10 minus 2 is
Okay, yes, we are getting 8 ATP.
so 8 ATP is in glucose to pyruvate. Okay, now we know that. Next, always remember now we have everything twice. So now like we know that when pyruvate was converted into like I'm not specifically asking like what is the thing from glycolysis or what is the thing from the Krebs cycle. I'm like in the entire like if I give you one molecule of glucose, how much ATP I'm getting. So in that way, we will include this NAD as well. This NAD conversion into NADH. So here we are getting, as I told you, like one pyruvate will give one. That means twice. So we have two NADH. That means 6 ATP from here. Okay. Now, from this acetyl CoA to entire Krebs cycle, and then to oxidative phosphorylation, we are getting 2 into 3 NAD. Like we like in last I think we learned now. We are getting 3 NAD, right? 1, 2, 3. 3 NADH.
Yes, sir.
And then we are getting one molecule that means twice, I mean 2 pyruvate means 2, sorry, everything is 2. So then we are getting 1 FAD and
Yes,
we are
sir.
getting 1 ATP, right? So basically everything is twice. Understood that, right? Like because we have two pyruvate.
Yes, sir.
So we have twice the amount of NADH, twice the amount of FADH twice the amount of ATP. So from here we have gotten this is 6 ATP. Now from this it is 3 into 3 into 2, 18 ATP, right? Like
Yes, sir.
3 into 3 ATPs and into twice. And here it is 3 into, sorry, 2 into 2, 4 ATPs, right? 2 ATPs. So overall here it's 14 ATP, and from here it is how much?
24, sir.
So what is the total outcome?
38.
How much? 38.
38, sir.
Yeah. So total one glucose molecule will give 38 ATPs, right? So this was, so now forget that. Okay, next is, so like many question, like many exams ask a like trick kind of question that if I have give like if I give you one glucose and if glycolysis is occurring, how many ATPs you have? So you always have to tell 4 ATPs, right? Because NADH is not, sorry, 2 ATPs. Glycolysis only gives 2 ATPs, right?
Yes, sir.
And if I ask you, one pyruvate can give you how many ATPs then?
20, 24
Yeah.
plus
Hmm.
6, 30.
30?
So because pyruvate is also converted to acetyl CoA,
Okay,
and then
from
we
here,
get
okay, yeah. So from here it is 24 plus 630, yeah. So one pyruvate, 30 ATP, and one glucose, 38 ATPs. And now, like this is for the, like I mean, based on the substrate. Now, if someone asks you about glycolysis, how many glycolysis, how many ATPs? That is only 2 ATPs. If they ask about specifically about Krebs, you have to tell that 24 ATPs, right?
yes, sir.
Okay. Now let's go to next step is oxidative phosphorus. Like you don't have any other doubt in Krebs cycle, right?
Yes, sir.
Okay. Now let's go to
Oh, sorry,
oxidative.
sir. Just one last thing. What is the meaning of biological tethers?
What is the meaning of?
Biological tethers.
Spell it. Biological.
TETHERS.
Never heard of it actually. Like, do you have any definition?
Sir explained something about it, but I couldn't get it.
Biological tethers. There is one or two carbon bonds. So what was the like? I mean, what was the definition that Sir gave? So I think like based on the definition that it is showing in here, like I think the complex that we studied, right? That pyruvate dehydrated complex. So based on the definition here, it basically says the carrier of a carbon compound. So here the complex, that is E1, E2, E3, the entire complex is the biological theta, because it is carrying a carbon compound from one enzyme to the other one. So I think that's what Sir would have given, because here it is said that it is a molecule, like a biological theta is a molecule that carries one or two carbon intermediates from one active site to another. So from one active site to another active site. So that's what the definition is given here. So was Sir mentioning the same?
So I'll give examples of three compounds, like the enzyme 2 we just studied, and then the pyruvate carboxylase and acyl carrier protein.
Yeah, this they do the same thing. Yeah, like here, I mean, based on the definition, I think like pyruvate dehydrogen complex, this complex is doing the same thing. Maybe that carboxylase will also do the same.
Okay, sir.
Oxidative phosphorylation. So what is oxidative phosphorylation?
So two things happen. First is oxidation of the reduced electron carriers, and then phosphorylation of ADP.
Phosphorylation of ADP in the presence of?
In the presence of downflow of protons, like downhill flow of protons. In the
Again,
presence of ATP synthase.
again oxygen. That's why it's
Okay.
called oxidative phosphorylation.
Yes.
The
Yes.
ADP to ATP. This thing is the oxygen plays a major role for in this. The NADH is coming to NAD. The proton goes here. FADH2, FAD. Proton goes here, and then in the presence of oxygen, the electron is it is helping in the form the conversion of ADP to ATP.